Thank you so much for having me. Today I'm going to be talking about Delta-8. Some of the earlier talks did talk about Delta-8, but more from a product perspective, purity issues, quality issues. So today I'm just going to talk just about the actual molecule itself, both from a scientific perspective, but also from a historical perspective. Here's my disclosure. So we've worked with some companies on the pharmacology of Delta-8 in the past, but really we have no ownership in any company that produces or sells Delta-8. So no real direct conflict of interest. I'm not trying to promote any particular cannabinoid. So interest in Delta-8 or awareness of Delta-8 is something pretty new amongst the general population. If you look at just internet searches for Delta-8 as a measure of awareness or interest, well, you see that before 2019, really nobody was searching for it. Probably nobody or very few people even knew what it was. After around 2020, you see that interest in searches just kind of exploded. And this plot goes up to 2021, but I'm sure that has just continued to go up. You see this plot is by state. You see the states where there's the most interest are actually those states in which Delta-9 still remains illegal at the state level, which isn't really surprising. But the fact that awareness of Delta-8 is fairly new has given some people the impression that Delta-8 as a molecule is new. And so I read pretty regularly quotes like this one. So this is a quote from an article in Forbes that was published just this year. So it says, Delta-9 has been studied extensively for nearly 60 years, but little research is published on Delta-8. Well, I don't think this is true at all. In fact, research on Delta-8 is not a new thing. It's been going on, as you can see from the title of the talk, for over 80 years at this point. So obviously not quite as much research as Delta-9, can't compare it to that, but quite a few studies. So we published a review article on this last year. So we pretty much looked at every single pharmacology study of Delta-8. There's well over 150 publications, not getting into the chemistry or anything, but just getting into actual studies of the pharmacology of Delta-8, over 150 publications. So I'm going to give you a quick overview of some of the findings today. So first of all, what is Delta-8? I think most people in this room probably know. Here's Delta-8 structure compared to Delta-9, and you see overall they're very similar. They're isomers, just one slight difference of the location of this double bond. So this gives it just a slightly different three-dimensional shape. So perhaps Delta-8 can fit into the cannabinoid receptors a little bit differently. Perhaps it can fit in a little bit differently with the enzymes that metabolize THC. Then there's also one additional layer of complexity. So when you take edibles or when you take THC orally, it's metabolized fairly heavily before THC reaches the general circulation. And one of these metabolites is actually active. So this is the 11-hydroxy metabolite. It's formed both for Delta-8 and for Delta-9. So when we consider what are the differences between these two molecules, we have to consider not just the differences in the parent molecules, but also is there any difference in these metabolites of 11-hydroxy Delta-8 compared to 11-hydroxy Delta-9. So just to give you a historical frame of reference, pretty famous year in cannabinoid research was 1964, where Delta-9 THC was isolated and its chemical structure was identified by Raphael Mechoulam, and Delta-9 was recognized to be at that point the primary active or psychoactive component in cannabis. But I want to go back even 25 years earlier than this. So 25 years before the isolation identification of Delta-9 was actually the first synthesis of Delta-8 and actual testing in animals and in humans. This perhaps isn't quite as well known, but this gentleman that you see here, his name is Roger Adams. He was an American chemist at the University of Illinois. And it's kind of an interesting story. So in 1939, the US government, this is 2 years after the federal government de facto made cannabis illegal with the Tax Act in 1937. Then in 1939, the US government had actually asked this chemist to isolate the intoxicating compound of cannabis. Well, we know he wasn't successful in that because we know that Mechoulam did it 25 years later. So what's one of the reasons that he wasn't successful? Well, the US government actually gave him samples of Minnesota fiber hemp to work with, which most people know has very, very little Delta-9 THC. So he wasn't successful in actually identifying this at the time, but what does fiber hemp have a lot more of? Cannabidiol, or CBD. So he actually published on the isolation of CBD in 1940 in a partial structural identification. So what does this have to do with Delta-8? Well, at this time, Roger Adams started playing around with the molecule, making derivatives of CBD. And one of the things he did was he put CBD in an organic solvent, he added a little bit of acid, and he heated it. That's a reaction that I'm pretty sure every chemist in this room is familiar with. It's how even today we're converting CBD into Delta-8. So this reaction was first done over 80 years ago. And so what he discovered at that time is that when he did this reaction, CBD could be converted to two different molecules. And he was able to partially identify these molecules and realize they were two chemically distinct molecules, he was able to partially identify the ring structure and had an idea that these were tetrahydrocannabinol molecules, but couldn't actually tell exactly what they were. But he tested them both in animals and realized that they were both psychoactive. And so he hypothesized that perhaps one or some combination of both of these molecules were what was responsible for the psychoactive effects of cannabis. Now we know today in retrospect that these are Delta-8 and Delta-9 THC, but at the time he wasn't quite sure what he was working with. Nonetheless, him and his colleagues decided that it was time to actually test this in humans. So the first clinical testing of Delta-8 was actually done prior to 1942, or it was published in 1942. And so this was done at Welfare Island Hospital. So Welfare Island is now known as Roosevelt Island in New York City. At the time, it had a prison and the penitentiary hospital on the islands. And so they actually did a clinical study using volunteer prisoners where they administered various different types of cannabinoids. One was just a crude cannabis extract, which of course has Delta-9. He also administered the CBD-derived Delta-8, and then there was also two other cannabinoids that he tested as well that I won't get into now. But given this was a study with 77 subjects, even to date, this is essentially the largest clinical study that we have had of Delta-8 THC, and at the time, they didn't even know what it was. So this was published in 1942. Actually, a very interesting article that basically, Roger Adams, subsequently to this study, decided that he wanted to do some additional studies, some additional testing. He sent samples of Delta-8 to about a dozen different chemistry colleagues. Then they wrote letters to him saying, hey, I tested it, I took it with dinner, this is what I felt. And some of those descriptions are pretty entertaining, so I can definitely provide the reference for people to read this article. This is pretty entertaining. But the effects of Delta-8 that were actually observed in this study in the prison were pretty familiar. So you saw elevated pulse rate, dry mouth, sometimes anxiety, sometimes euphoria, increased hunger, drowsiness, laughter. And for those prisoners who had been high before or used cannabis before, they said this feels a lot like using cannabis. So this was the first indication in humans back in 1942 that, it looks like Delta-8 is acting an awful lot like Delta-9 or like cannabis, even before they even knew what Delta-8 actually was. After this, it was a pretty slow period for cannabis research. After 1942, U.S. entered World War II. The research priorities of the U.S. government just changed. Roger Adams essentially went on to retire from cannabinoid research. He worked on other things, and it was just a slow 25 years or so for cannabinoid research until 1964, when Meshulam identified Delta-9 THC, and shortly after, the structure of Delta-8 THC was identified. And now they really had the tools to assess Delta-8 versus Delta-9 THC quantitatively and see, is there any quantitative difference? Well, they started giving Delta-8, Delta-9 to animals, to different species, and they could clearly see, well, it's overall doing the same thing. Qualitatively, we see the same effects as cannabis, Delta-9. But when they started looking at individual species, they started to see some differences. So they tested, these are the very early animal studies in the 1960s, early 1970s, tended to be on monkeys, on dogs, on rats. And what they saw was a pretty consistent trend that Delta-8 was always a little bit weaker than Delta-9. So this is the first time they had really observed this. So in monkeys, you needed about a fourfold higher dose of intravenous Delta-8 to have an equivalent effect to Delta-9. In dog and rat assays, you needed about a twofold higher dose. So it was starting to look like there was a consistent pattern here. But over time, researchers started using monkeys and these dog assays less and less, and started using mice more. So they developed these, it's called tetrad assays. It's a set of 4 assays they typically do in mice. And if these assays have positive results in all 4, that usually indicates it's a cannabinoid agonist. So when they tested Delta-8 in these tetrad assays in mice, they actually saw something a little bit different from the other species. They saw that Delta-8 had the same potency as Delta-9. So that was a little bit weird. They didn't really know exactly, or I imagine they didn't really know what to make of this at the time. But keep in mind that at that time, they had never tested Delta-8 in humans, except for that one very early study where they didn't really know what they were testing, so they didn't know. Would humans look more like mice? Would Delta-8 be the same potency? Would they look more like monkeys and have very different potency? At that time, they just didn't have any data to say, or at least no published data. So they didn't have to wait too long. In 1973, they actually published, there was 2 studies published almost simultaneously, one from a group in the US, one from a group in Brazil. And between these 2 different publications, they actually tested intravenous dosing, oral dosing, like edibles, and smoked as well. Both of these studies compared both Delta-8 and Delta-9 THC at a range of doses, and so I just kind of replotted the data from all these studies. You see on the y-axis, essentially just a scale of subjectively how high people felt or how strong they felt the effects were. On the x-axis, you see different doses. So overall, what you see across these different routes of administration is very consistently across all different doses. At any one given dose, the effects of Delta-9 are almost always stronger than Delta-8. And while, of course, we kind of all know that's true today, at the time in 1973, this was like totally new data because people really didn't really have any idea at that time. So now they knew it was different. They knew Delta-9 was more potent than Delta-8 THC, but otherwise they knew it was doing about the same thing. But how could they explain this? Why is there this difference? Well, they didn't really have any ability to test this because in 1973, they didn't even know how THC worked. There was one hypothesis that there was a receptor for THC. There was another hypothesis that there was no receptor, that THC molecules were just kind of perturbing the neuronal membranes in a very nonspecific manner. So they had to wait another 15 years before they even identified that the actual mechanism and the specific receptor for THC to actually start looking at the mechanisms of why Delta-8 and why Delta-9 are different. So it was 1988, they first identified in the rat brain the cannabinoid CB1 receptor, and then shortly thereafter, they cloned the receptor, meaning they identified the actual DNA sequence. They could put that DNA sequence into cells and cause the cells to produce the receptor in order to study it better. So now 15 years after the first clinical data, they finally have this ability to actually see, okay, at the cannabinoid receptor, is there any difference at the level of the receptor of what Delta-8 is doing? And it turns out that every species has a slightly different version of the CB1 receptor. The human CB1 receptor isn't equal to the mouse CB1 receptor. It has a different DNA sequence. It functions slightly differently. So they actually did these Delta-8 versus Delta-9 tests using cannabinoid receptors from all different species. So, for example, rats. First, they did the rat receptor. Well, as I had said earlier, in vivo in whole rats, they saw about a 2-fold potency difference. And sure enough, looking in vitro effects of Delta-8 and Delta-9 at the rat CB1 receptor, it looked like Delta-8 had about 2 to 3-fold less affinity for the CB1 receptor, about a 2 to 3-fold difference in the potency and its ability to activate the receptor. So this seemed to make some sense that this is going to explain, this difference in affinity for the receptor is going to explain why Delta-8 has a different effect in vivo in rats. Well, they repeated this in mice. And remember, mice seem to be a little bit of an outlier that in vivo in mice, Delta-8 had about the same potency as Delta-9. I mean, on average, there's probably over a dozen different studies where they tested it in mice, and there's a range of results, but on average, it was about the same potency. And so sure enough, when they looked in vitro at the mouse version of the CB1 receptor, they saw that Delta-8 had about the same affinity, the same potency as Delta-9. So it seemed like this difference at the receptor was really what was driving the difference in whole animals. So that seemed to make sense. But then the next piece of data came out a little bit, I would say, unexpected, which was the human version of the cannabinoid CB1 receptor. So we know in humans, everybody has their own idea of what the difference in potency is, but let's say on average, give or take about 2-folds. And yet when they looked at the human CB1 receptor, they actually saw a 6-fold difference in potency. So Delta-8 had 6-fold less affinity than Delta-9, Well, that's kind of weird because we don't see a six-fold decrease in potency in humans. So I think this is something that's not 100% understood exactly why there's this contradiction between what we see at the human receptor and what we see in vivo. You could argue, well, it's different experimental conditions because different labs have these different conditions in which they run these assays, and perhaps it's just not a very accurate result, but 3 different independent laboratories have published the six-fold difference, give or take, between Delta-8 and Delta-9 at the CB1 receptor. So there could be some other mechanism going on here that we just don't understand yet. So finally, to finish up, I want to talk more globally about potentially other differences in the effects of Delta-8 versus Delta-9. Well, one possibility, maybe kind of an uninteresting possibility, Delta-8 does the exact same thing as Delta-9, it's just a little bit weaker. It's possible. Clearly there's a lot of overlap, but is it doing the exact same thing? Maybe. Another more interesting possibility is that it actually has distinct pharmacological effects that actually differentiate it from Delta-9. So one example of this, that at this point I would say it's just a hypothesis, we don't know for sure, it hasn't been tested in clinical studies, but some people report that Delta-8 produces less anxiety than Delta-9 at a dose that otherwise gives equivalent overall effects. So that's one possibility, and there could be other differences in subjective effects or therapeutic effects, and we just don't know yet. There's just not enough research. There is currently 1 or 2 ongoing clinical studies, which is good to see, but overall we have very little data to actually see if there is perhaps a case where Delta-8 could have a different therapeutic effect than Delta-9. So hopefully we'll see some more research. But is this even something that's likely, or is there a mechanism that could explain why Delta-8 might be different than Delta-9? Well, I think there is mechanistically a possibility for this. An example of this potential mechanism of how Delta-8 could be different is at the CB2 receptor. So I already told you Delta-8 is weaker at the CB1 receptor than Delta-9, but Delta-8 has the same potency as Delta-9 at the CB2 receptor. So let's just imagine a hypothetical scenario. This is really just me all hypothesizing. Well, the CB2 receptor, it's expressed very heavily in immune cells. It potentially has an anti-inflammatory effect, a very different effect from the CB1 receptor. So let's imagine this hypothetical scenario, which is just based on in vitro binding data. So on the left, you have Delta-9, and according to this data of this particular publication, 18 nanomolars of Delta-9, that concentration will produce about 50% binding of the CB1 receptor, which means out of all the CB1 receptor, half of it is bound to Delta-9. Well, CB2 receptor binding affinity is about the same as CB1 for Delta-9. It's maybe, according to this publication, a little bit weaker. So where you have 50% occupancy at Delta-1, you have about 30% occupancy at CB2. Well, now let's consider the situation of Delta-8. What's one thing we know? People will titrate the dose to the effect they want. So if they want to feel a particular effect of Delta-8, they're just going to take a little bit more of it compared to Delta-9. So let's assume the person takes a slightly higher dose of Delta-8 to compensate for this slightly weaker effects, and still they end up with 50% receptor occupation at the CB1 receptor. Well, because now they're into a higher dose range, and because the CB2 affinity is the same as Delta-9, they would actually end up with something more like 85% occupancy of the CB2 receptor. So if you're kind of lost with what does all this receptor occupancy data mean, let me just put it in the more simple clinical terms. At doses which give equivalent psychoactive effects, Delta-8 could have, at least theoretically, a greater anti-inflammatory effect compared to Delta-9. This is just kind of my hypothesis. This has not been, clinically proven. Nobody's directly studied this yet, but I just want to provide this as one kind of hypothetical example of how Delta-8 may have different therapeutic properties compared to Delta-9. Obviously, there's going to be a lot of overlap between the two, but perhaps there's cases where one could be more appropriate for one patient and another more appropriate for a different patient. So I'll end there. Thank you for listening. We did publish this paper on Delta-8 in British Journal of Pharmacology last year, so if you want to learn more about Delta-8, you can read that. It's open access. Otherwise, I'm happy to take any questions now if there's time, or later you can always just send me an email. Thank you.
Thank you. I look forward to reviewing your paper. A question, any idea of the differences between Delta-8 and Delta-9 at other clinically relevant targets like the PPARs or the TRP channels?
They have, to the best of my knowledge, and this was a year ago, we reviewed the literature pretty comprehensively. So unless it's something very new, they haven't assayed at any other targets besides CB1 and CB2.
Got it. Thank you.
Okay, so I'm getting kicked off stage, so thank you for listening.